So we've got three great speakers, and I'm going to set the scene first and really try and look at how we merge things together and get the silos throughout the supply chain to operate in synergy so that we can connect therapies to patients. But there are two things I want you to have in your head as we go through this. The first thing is this is a workshop. The workshop only works if you ask questions. So at the end, when I ask if anybody's got any questions, could somebody please be brave enough to go first? Otherwise, the tumbleweed blows past and it gets very painful. The next thing to have in your mind is the concept of thinking about the future. So what's your commercial scale supply chain going to look like, and how are you going to build, test, and optimize to get there, so that we don't pop out of a pivotal scale trial and then suddenly realize you can't distribute globally. So with those two things in mind, I'm just going to set the scene. The structure today will be everybody, or the three speakers, will give a 10-15 minute overview, and then I'll bring them all back up on the stage, and you'll all have some fantastic questions ready to go. So with that, sort of the hypothesis or the idea that led to this panel was, if you thought the first car was actually sold in 1886, so you could argue that was a commercial business. By then, people were buying cars, they were driving them around. But it wasn't until Henry Ford created the production line, amazingly this week, about a century ago, that actually an industry was created. And I think you can draw the parallels to that to where we are with advanced therapies at the moment. So everybody's talking within manufacturing about closing and automating it and sort of locking all the unit steps together. If you pull that up to the macro scale, and you have a very simple supply chain of donor hospital passes it to Courier, Courier passes it to manufacturing, manufacturing to Courier, and then to your final therapeutic hospital, they're all collecting broadly the same bits of data. They're managing the same cells, but none of them are talking to each other. So I think how do we pull those silos together, and how do we integrate the manufacturing, the logistics, and the clinical platforms? So to take us through that, the Rogues Gallery, we've got Phil, who talked to us around the manufacturing piece and how you interact with the logistics platform, for example. Dominic will look at what we've termed the first 100 meters. So how do you collect that apheresis, that raw material, and how do you feed that into the manufacturing platform? And then Jackie looking very much at the last 100 meters of these things arrive at a clinical setting. They're in a dewar that has a meter cubed sort of footprint. How do you move that around a hospital? How do you get that therapy to a patient without an uncontrolled thought? So just to throw some ideas out there to try and seed some questions, there are four things that I think are driving complexity at the moment. So firstly, this is ARM data. I've just mapped out what they've put out every quarter. And there are more and more therapies, but everybody's doing it in a slightly different way. So it may be a cryopreserved therapy that you thaw and then infuse, but everybody has a different system, everybody has a different packaging system, everybody has a different label printer. So how do we drive standardization into this to help the clinical process? If you look also at the number of clinical sites, now this is our data, I've put this up before, we're delivering to about 55 countries every year regularly with advanced therapies. And that's probably going to flatline out. It's a $500,000 therapy, it's a first world treatment at the moment. But what we are seeing is massive growth in the number of clinical sites that are treated. So there's now over 2,000, and I need to rerun this in a couple of months for the 2019 data. But if you've got 2,000 different sites, that's 2,000 different nurses, 2,000 different SOPs, 2,000 different ways of storing it, and I can go on and on and on. The other way of looking at that is each of those clinical sites is now having to manage more than one therapy. So when you started, it was a CRO bringing one therapy in a month. It's now 5, it's now 10, and it's growing, and they're all done slightly differently. So how are they actually going to manage that complexity within the clinical setting? So we need to think about how we help them with that. Also, there's volume going out. Now, these are our CAR-T shipments. Obviously, I've anonymized it, but green is basically what we've done to date, and then red is the forecast going forwards. And these are now being distributed on a commercial basis into 31 countries globally, so the growth is spectacular. But everything's done sort of manually. It's terrifying how many Excel spreadsheets are used within a single supply chain and how many manual entries. And basically, if something's going to go wrong, it's generally because the person in there has done it wrong. The biggest error is generally human. And we also need to think that there's an evolving technology. Now, this is just looking at cryo shipments. So in 2014, about 4% of all of our cell and gene shipments were cryo. It's now 25% and growing. And that's sort of pushing control into the supply chain, but it's also changing what clinical sites have to manage. So my question is complexity coming from that, or is it just coming from a lack of coordination? And if we all talk as a group, as a collaboration, maybe all of these challenges would be addressed. So to throw 3 more points at you, so aligning platforms, sort of the title of this presentation. So this was data that came from a paper, and it's just a process flow map. And I think we can all agree that every step in here is a critical action, and failure could be catastrophic to the patient. It also means in a clinical trial, you might not get the data you wanted to generate, or at commercial scale, you wouldn't get reimbursed. So you're either upsetting your stakeholders or your shareholders. But if we just very simply overlay red for clinical platform, green for logistics platform, and blue for manufacturing platform, you can sort of see the interrelations between them, and we need to make sure that we're not just throwing it over the wall from silo to silo and integrate that. If we look at it another way, and I know this is turned up really, really small, but this was actually mapping things from London and then shipping out to various countries in the world. And the 2 things I've tried to highlight there are actually the customs transit times. They're different. You know, most countries, 2 to 4 hours. China, you're looking at 48 hours, and that's just their process. Just deal with it. So how do you manage that? If you've got a 24-hour shelf life, how are you going to manage those kind of things? And everywhere is different. If we look at that in a different way, we've just lane mapped here from the East Coast US into Amsterdam, London, Berlin, Paris, and then Milan. And you would have thought actually those cities coming from the East Coast would be broadly the same transport times, but they're different because planes have to route. Some of them aren't direct flights. Some of them have customs agencies that are slower than others. The other thing that's interesting is if you do a 9 o'clock pickup or a 12 o'clock pickup, your transit time changes. So you don't just look at the duration of the flight, it's when does it go, which is best, which are the directions. So we need to think together because that would inform your manufacturing strategy. And then finally, I've sort of had this rant before, but around hand carries and what impact that very simple decision can have on you as you go forwards. So if we think about phase 1, there's an academic, brought a great technology, he's got their first investment. They want to use hand carry, which is somebody sat with a box on their lap on a plane, because it's reliable and you feel safe. There's a transplant lab just down the corridor, so they use the organ box to do it. It's been validated, they know it'll hold temperature. Great. They go through phase 1, everybody's happy, they get more investment, they move into phase 2. And now everybody's focusing on reimbursement strategies, closing and automating. How do they get the next funding round? And so actually logistics just carries on the way it carries on, and people tend to throw resources at it. So there's a cost in there and a complexity that you don't see because people are just doing it, and you've got bigger problems in your world. However, then you then hit phase 3, or apparently phase 1 again, according to this slide. And you suddenly realize that actually there aren't enough people to carry enough boxes on enough planes for you to be able to scale up and commercialize. So you then start thinking, well, we'll ship it through the hold of the plane. It's fine, it's done all the time. But the packaging system you've got can't be stacked, so you can't put it in the hold. Now you have to revalidate a new packaging system, and you have to work out a new paperwork trail to put it on the same plane, but sort of below the passengers as opposed to with them. And maybe you have to go back a year. So you need new investment, you're slower to market, maybe you don't get your first to market designations. So those tiny little things need to be thought about, just to emphasize the point that we need to work together. So with that, thank you for your time, and I'll hand over to Phil to look at the manufacturing side.
I think nobody in this room is a stranger to the complexity that this industry throws at us. I just wanted to kind of talk a little bit about some of the manufacturing challenges. Now, it's a very broad topic, obviously, so I will try to drill in. Looking for, there we go. Push it once. All right. So I think all of our collective goals are to get these therapies to the patients that need them in a safe and effective way. I am going to be making some forward-looking statements, obviously. This slide just tells you that I'm making most of this stuff up as I go, so, But I like this as a starting point. I mean, one of the things we talk about, and there's some companies represented here that are doing really innovative things around modifying cells, engineering cells. And I've said, 5 years ago what I thought would be a provocative statement, saying that within 5, 10, 15 years, every cell that was manufactured for clinical purposes would be genetically manipulated or modified in some way. And this is a paper from Peter Zanstra and a team that came out a few years ago now. But I sort of like this projection as, hitting the wrong button, as people, you won't be able to see this, but you can sort of characterize the evolution of therapies, whether they're originally from natural products, evolving all the way up into more complex small molecules, larger molecules, and biologics, monoclonal antibodies, ultimately leading into the upper left-hand corner of the chart on the right-hand side of the slide. These cells are obviously, the cells, the genes, the vectors, whatever these components are highly complex, large macromolecules that have a significant challenge not only in the manufacturing, but as Simon alluded to, the distribution, shipping, and dissemination and administration is quite a bit more complex. But I like this, and more you're going to hear people talking about kind of the not reprogramming of cells in the context of iPS cells, but sort of, I used to say, installing a new motherboard into the cell or repairing the genetic componentry through engineering. And that adds a significant amount of complexity that Simon was talking about. So that's one component or one part of this complexity. So if we think that, and I know that Dominic's going to talk about sort of the highly heterogeneous starting material that we also deal with, whether it's for donors, allogeneic, or whether it's a patient-specific autologous therapy, but the biology is highly complex. And on top of that, we're starting to engineer it, which adds additional complexity. And then the challenge in biology, I see, partly is we don't know enough about, we talk about machine learning and artificial intelligence being applied to biological systems. That's all well and good, but you need very, very strongly curated data and correlative data to be able to understand what the biology means. So those of you who know me have heard me say this before. We're still in the days where you poke the frog with a needle and its leg sticks out, and we go like, that's really cool, that's an effect, I like that. And then we go, well, why did it happen? We go, I have no idea. So the biology is changing, and that adds complexity to those of us in the room who are manufacturing technology providers, because we have to anticipate what the manufacturing processes and facilities are going to look like in 5 or 10 years when these products start to scale. So I'm going to set the biology aside for now and start to talk a little bit about the process or the manufacturing part. And the process, these are the challenges we talk about. Do you centralize manufacturing, or do you go with decentralized? Do you do your own manufacturing facility or build your own manufacturing capacity, or do you work with a CDMO? At what scale? How many patients do you anticipate treating? Where are they going to be located geographically? How do you get them to those patients in real time? And most importantly, how do you actually track and control the process, which becomes that third element in this ecosystem, which is the logistics. And that was something that Simon introduced us to. And there's a term, I know some of you who have heard this from other speakers as well. We talk quite a bit about chain of identity and chain of custody, but implicit in that is this chain of quality, making sure that not only the product gets to the patient, but that it is exactly the product that was manufactured wherever it may have been manufactured. And the logistics is really an element of connectivity and control. And then down here in this little box here, you may not be able to see it, digital. The digital wraparound that makes all these things not only work, but then ultimately connects them, and then connects each of these elements into the broader context of the clinical setting and into the delivery process that follows. So I'm going to dive down and talk about one part of this. I could talk about lots of different parts about the manufacturing process, closing systems, connecting them, digitizing. I'm just going to talk a little bit about one in the interest of time, and then sort of to foster or seed some additional questions. But the challenge with all of that complexity in the ecosystem and the evolving therapies and what all of you as therapy providers are doing to us as tool and technology providers is the need for absolute process validation, the ability to say that the process is effectively the product. Now, I hate that statement. The process is the product is the process. Unfortunately, today, because we don't have or we have an evolving landscape of effective potency assays and cell qualification assays and comparability assays, we are dependent on controlling the process and letting that process be a proxy to indicate that the cell product that is going through that process, notwithstanding the heterogeneity of the patient population, but the process is controlled and we understand that. So one of the challenges there is the data integrity. And data integrity, if you look at this chart on the left-hand side, FDA warning letters citing solely data integrity has been increasing dramatically. Now, is this because we're collecting more data? Is it because we are working in a highly complex environment where we're populating data records by hand in a clean room? Those of you who work in GMP facilities in manufacturing, those days are long, they're hard, they're not very comfortable for some of the operators. So it does lead to an increasing amount of potential error there. So this slide is just kind of highlighting that one element of the data integrity. If I jump to the next slide quickly, these are extracts from recent FDA letters. On the left-hand side for you, handwritten records. Like I just said, if you're in the clean room, you're gowned up, got gloves on, and if you've got paper records that you're writing, you've got an observer behind you. You know, early in the morning, your handwriting might be neat. You might have had a cup of coffee and start getting jittery and wobbly. But if you see, and I don't have the slide, I took it out of interest of time, but there's a lot of heterogeneity in how we write, and people writing batch records. Sometimes a decimal point looks like a circle or a smudge. Sometimes it doesn't show up legibly. Sometimes a 7 looks a lot like a 1 for some people. So there's a lot of inherent human error in writing down. You'll notice a couple of these things. Using whiteout and writing over and not initializing changes. I mean, that's bad training or just a poor adherence to the practice of cGMP manufacturing. But it's not just paper. On the left-hand side, I mean, this is an example where you're using Excel. It's not locked. It's not controlled. The formulas in the cells are wrong, so you're miscalculating things. And these things can pervade the entire manufacturing process. So there arguably has to be a better way. So we already, as an industry, collect a lot of data. At the instrument level, every piece of equipment has the ability to not only be controlled through the user interface, but it also has the ability to export data, whether into an Excel file or into another data format, which can then be used for either analysis or for just electronic batch records and for ESOPs and the like. So every piece of equipment has a little bit of a different way of doing this, which adds complexity. And then when you start to integrate into a facility which has multiple third-party vendors, that adds another layer of complexity. How do you federate that information so that it's not only available to the manufacturers, but how does it populate a QMS, an EBR, and sort of how do the operators have access to the ESOPs or the SOPs that they need? So we have endeavored to start to build a capacity from the ground up. You know, there's other people in this at this conference who are also doing similar things, and you know, we're not saying that this is the end-all be-all as a manufacturing execution system, but it's a foundational part. It takes and collects information, feeds it up into the cloud. It's protected and controlled. But more importantly, what we really were trying to focus on is what is the manufacturing environment for an operator in a clean room, and where are the error opportunities to be removed from the system. So, you know, simple things. ESOPs need to be captured. Who, what, and when is operating, and what are they operating? You know, barcodes so that there is no handwriting and interpretation of handwriting. Formula which can be sort of approved in advance. The operator doesn't have the ability to change that. They just have the ability to scan and pull something in from the data. And then ultimately, all of that information, we're collecting it. Why wouldn't we use it to improve the processes that we were talking about earlier? So the ability to look at operational and get operational insights in real time. You know, environmental control, environmental monitoring. It would be really nice to know, hey, we've got somebody touched something they shouldn't have touched. Their hands were dirty. We're getting a real-time readout. And then ultimately, this becomes the opportunity for us, if the process is completely controlled, all of the electronic batch records are controlled in a quality management system, you have the ability to move to the future where we could release products by exception. So you're only looking for deviations in a process. If everything goes swimmingly in that clean room and the operator has logged and collected that information, suddenly you get to a point where somebody can look that somebody outside the clean room can look at a record that has been entered, say, yep, I consent that this is okay, they sign it, and then off they go. So this is the platform. I'm not here to sell this to you. If you're here to buy it, great, but I'm not here to sell it to you. Just to talk a little bit about kind of this approach and this way of thinking about things is that we have to take out manual labor. We have to have equipment talking and automatically populating the information into the sort of the digital ecosystem. And again, that provides us the opportunity not only to remove the likelihood of error being introduced by the operators, but ultimately it gets us to the analytics. And as we start to better understand what's slowing us down in a process, where are the risks inherent in the process, whether it's at the handoff to a logistic and supply chain, where are we seeing these issues crop up? These types of tools will be absolutely critical. So with that, I'll pass off just to say two more, this slide and one more. The manufacturing process is highly complex. And you've heard us and others like GE talk a little bit about what is the manufacturing platform of the future. Well, we think it's just closed, it's connected, it's digitized, automated, scalable, has to be highly efficient to take cost out. But that on its own will enable that release by exception I talked about. Data analytics, real-world data and real-world evidence, network orchestration, machine learning, reduced complexity, and then ultimately a set of standards that we can adhere to and apply to, not only electronic standards, but process standards and the like. So these are some questions, and I left them in. I was going to take this slide out because it actually comes from our bioprocess colleagues in our business, but it's the same problem we face. So this is not a cell and gene therapy manufacturing challenge. It's a complex ecosystem manufacturing. But you'll notice we're focused on a few key elements. Speed to market. How do we help this industry get the products to patients? How do you do it efficiently with agility and the ability to adapt to changing environments? What's the optimal manufacturing process, especially as we start to globalize the industry? And supply chain. Is the supply chain robust enough? And with that, I'm going to hand it back to Dominic. I'm going to hand it straight off to Dominic. So thanks for that. I know we're going to have questions coming up, so thank you. Yeah, thanks.
Let's see if I know how to make sure I know how to work this. Thanks, everyone. I appreciate you all joining us here today, and thanks to Simon for organizing this great session. Phil certainly alluded to a number of key challenges that we have within the industry as we scale up. Obviously, the analytics and everything speaking to each other and just connecting it from start to finish. I'm going to take it back to the beginning now and talk more about the front end of this entire process. And I just noted the industry as a whole has a challenge of obviously standardizing terms. And as I just found, Simon, you talked about the first 100 meters. You'll see in my terms I have the first mile. So we can't even get it coordinated here. So I'm going to focus on the starting material, whether it's obviously there's the challenges around managing the apheresis from the clinical standpoint. I'm coming from a perspective with Hemocare around the actual donors and managing of the donors. So a lot of what I'll talk about is focused on the donors, but it applies to the patient or the donor from this standpoint. So I threw this slide together and I was kind of looking back at some of the history of the industry and where we are today and where we were. And I pulled out some different excerpts from papers that are out there in the literature. And looking back around 2011, the industry obviously was a lot smaller, but there was a lot of excitement. It was right around the time that Dendreon got their approval. And so looking at the far left side, I think this does it, I put in three key concepts that were coming up at that time. We've always been talking about manufacturing challenges. That's been the major focus from day one. How do we manufacture these products? Certainly achieving quality and consistency. This was around the actual cells at the time. And then down here, stability and logistics. I remember at the time, Dendreon and their final product was, it was stored 2 to 8 degrees C. So you only had a short amount of time from a stability standpoint to get that product from the manufacturing site back to the clinic. So now fast forward to, generally now, although this is actually a little bit behind too, Same challenges, difficulties in achieving consistency in your process controls, as alluded by FDA and also just in what Phil just talked about. Capacity challenges, back to the manufacturing. So we're working on manufacturing, but now we have challenges and where we're going to get it done. And certainly where we are today from a supply chain complexity, as was just alluded to. Back here, again, back in 2011, we were really focused on the final product. We're still focused on the final product, but now the bigger challenge may be the front end of this whole process and managing all this patient material. So I think it's interesting that where we are, we're learning, we're getting better at this, but as again, the challenge is the scale. So I pulled this information from the Beacon source from Hanson Wade, and so this is looking at just the CAR-T and the TCR trials that are out there, and you can see a total of 791 clinical trials to date as of June, and interestingly, you can see where the majority of them are in the early phases, but this is the kind of the two numbers that really kind of stuck out at me. Right now, they're tracking just about 3,500 total patients that have been enrolled for these 791 trials, but if we were to enroll all of them, that number would be 76,000. So, and again, most of them are autologous now with the allogeneic coming to this, but so now, again, it goes back to the scale. How do we manage, if we're struggling now to get processes in place for 3,500, what does that mean for 76,000? So just taking that a different way. So again, right here is where we are today. As we begin to scale this up, hard to say how quickly that's going to happen and what we do with it, but certainly now is the time to really develop those scalable systems and put them in place, especially for that first mile or 100 meters, so that we can manage that patient enrollment or donor processing and so forth. We've seen many varieties of this particular schematic of looking at the process, but again, from start to finish, it's an integral process, and if we now bring it into where the importance of and the role of the starting material, the starting apheresis material, again, whether it's collected at the clinic or at a donor apheresis center, it all starts with the challenge of not only the inherent material, but also, not ready for that one yet, but again, the material itself is inherently variable, but then you're also predisposed to the challenges that come with the patient or the donor. Every one of us is different. So once you start putting that variability in here, it's hard if you don't have a robust process in place to go from this point to this point and have a consistent product. And so, certainly it's priorities early on in the process to truly understand what you can and cannot control in that process, so that way you can begin to propagate at scale. So talking about the variability and the challenges now in managing that starting apheresis. So I've laid out, I spoke with my team and really kind of dug into some of the details that is involved in, and again, this is focused on an apheresis donor, a normal healthy donor, which has its own challenges, of course, but think about that with a patient now in a hospital, but walking through this, it takes a lot of effort to ensure that when someone like World Courier finally comes to pick up the product at our facility, any number of things can go wrong, and that impacts everything downstream. And this is just, this process is just one donor. So, and again, the first step is really just, you got to locate the appropriate donor or the patient. Challenges that come with managing that donor to make sure that they actually are engaged, that they're going to show up when they're supposed to for that collection time. Then it's the actual collection itself, and arranging to make sure that the logistics are set up, because once you do the collection, it's usually fresh. And so the time that product is collected, you have just a short amount of time to then ensure that the couriers are there to pick up the product, to make it onto the plane, to get it to where it has to go. So, a number of things there. Of course, all the batch records have to align to keep that traceability. Then the apheresis comes into play. Everything has to go as planned with that as well, You might have everything up to that point, and then the donor is on the bed, ready to collect, and they ate something or they drank something or they didn't drink enough, and the flow of that collection changes. Did you build in enough time for that? From that point, once it's collected, now again, the logistics comes into play, which we'll highlight some more later. There's so many different logistics players and so many different aspects that come with that. There's times and there's questions. Who's going to own the pack out? Is it our team? Is it the clinical team? Is it the logistics team? If we're doing it, do we have our pack out pre-primed and ready to go? Is it pre-chilled, or we're relying on the courier to come with that? What happens if they don't bring it? So there's challenges in that piece, but then all that has to happen, and then finally you get into the bigger challenge of perhaps getting it from that point to the manufacturing site. So thinking about all that, ultimately now, some things that are challenged with, especially again, now this is just one, if you're just thinking about one person, one patient, one donor, but again, what does this look like when it scales? Just with the patient, you can't obviously have a backup for your patient, but from a donor standpoint, the collection, the donor's late, if they don't show, if they don't pass the day of screening, if there's incorrect or missing documentation, whether there's a collection. So, at least from a standpoint of an allogeneic process and a normal healthy donor, you can always think about having a backup, and then oftentimes you have to have a backup to the backup just in case things happen and you want to get that product on time. Certainly from a logistics standpoint, we just talked about them. The courier being later perhaps doesn't show. Shippers aren't preconditioned. The flights get canceled, as they often do. Missing or incorrect documentation that, again, if it's going international, can't get through customs, it sits there. What do you do? At the other end, everything was set up, there were delays, now the product gets to the manufacturing site and nobody's there. Now what do we do? So again, it's a challenge from just one patient or one donor, but as we saw the scale, how do we really put the processes in place to ensure that we're ready for the growth? Some of the things that the industry, some of the things that we're doing, one of the prime ways to consider, as they've done for the final product, is moving away from the challenges of fresh and the limitations on stability and thinking about cryopreservation. It's happening more and more. Certainly from a starting material standpoint, if you think about the patient material, how do you manage all that patient material? You don't know when they're going to be able to collect, so cryopreserving at the point of collection, whether it's the patient or the donor, obviously enables more, the material management, certainly preserves the quality. Obviously, there are some challenges that need to be considered from a cryopreserved product, but preserving the preservation and stability is a key aspect to providing flexibility downstream. It avoids some of the shipping and logistics delays. If it's frozen, it can sit for a period of time. Downstream processing consistency, certainly staff and project scheduling and staffing, and then supply allocation. Another aspect that we really focus on at HemaCare, from a donor standpoint, is ensuring that if you have a donor that you're relying on to support, you've got a client that really wants a product, you need your product, and you want to ensure that that product's going to consistently get there. One, if you have donors that are reliable and recallable, that they're there when we call upon them, they're willing and able and ready to go, so you're not hoping that they're going to show up. We know that they're going to show up. Again, that also is a product. If you want consistency, you can always go back and recall that same donor to get the same material. Again, for an allogeneic standpoint, it's really critical to be able to have that type of consistency. And then having a wide breadth from a donor pool to really actively go out and find the products and requirements that you need. Another aspect, again, going into the scale up and the clinical demands of the products. We built our new facility that also now has clean rooms and GMP processing capabilities right on site. So what does that do? Certainly it allows us to do the cryopreservation immediately post-collection, which again gets you the freshest possible product processed immediately. It also enables that product to be processed on site to support clinical requirements as opposed to having to ship it fresh to some other facility where then you start to lose some of that control. So, as with Phil, I threw up some questions here that I think that we're challenged with on a regular basis at HemaCare within the industry. I think one of them that I let Simon in on so that he was prepared so we can answer this one together. You know, with all the specialty couriers and logistics platforms that exist, a challenge for us at our site is which one do you go with? You know, how do you standardize when there's 10 different logistics options, 10 different logistics platforms? You know, we have to go with whatever our clients say to use. Well, we want this one. Okay, well, we can do that for you. Someone else wants this one. So it's not just us. It's not just, you know, the world couriers or any of the other suppliers. It's the industry as a whole as to how do we stop being quite so customized or individualized and come up with a path forward that can be a little bit more standardized. And it's certainly possible, and we're going to do that, but that is certainly a challenge that we deal with every day. Again, making the materials more available to ensure process flexibility. Effectively standardizing methods. There are methods out there or paths to standardize the collection process, but then there's the processing. And again, there's numerous ways to process a product. For us, you know, again, thinking about the industry, where the industry is going from an allogeneic standpoint, how do you manage the long-term demand for unique allogeneic donors? If everybody wants their own specific donor and they want to keep them for themselves, sooner or later there may become a limitation or a challenge in managing all of that from an analytics standpoint. And then can we develop a robust supply chain to support the therapeutic uniqueness while still meeting the necessary global flexibility? So that I will pass it over to.
Hi. Simon asked me to talk about the last 100 metres or last 100 yards, but I'm actually going to do both because the donors in this case are the patients which are in the hospital, so that's the first 100 metres or yards, and I'm also going to look at the delivery back into the hospital setting. And I'm going to talk, I'll talk about some of the challenges that there are for these products, which I know everyone's heard again and again in this conference. But I'm also going to talk then about what we're doing in the UK, and we've got an initiative to try and tackle these. So this is a really simple schematic that I came up with myself instead of the really fancy ones. But what I want to show here, and it's a really obvious thing, is you may have a couple of hospitals involved, you may have some apheresis units involved, and then you've got a manufacturer, and you need logistics between all of those. You then may add in, we talked about a cryopreservation step, so it may go off-site to another cryopreservation step. And what we've heard again and again, even with people like Novartis and GSK, you know, big guys, they're saying, this is really complex, it's a really complex thing to manage. And there needs to be a flow of information, and that needs to be really well handled. So if you think about the clinical sites, you need to schedule in your patient. And if you think trying to get a donor is difficult, if you've got a patient who's been undergone immunosuppression, they're not well, you know, you have to really work well with the apheresis site. You then have to ensure that the patients are managed, that they are appropriately treated, whether that be before they have their sample taken, which then goes on to manufacture, or they're prepared for the manufactured product to come back. You have to have decent trial management. If it's in clinical trial, then it has to be managed really quite carefully, and this has to be coordinated with the manufacturer and with the logistics provider. And then you have the difficulty of the pharmacy and the cell lab, and which one of those is the primary treatment locale for receiving the product and then treating the product, preparing the product. And then you have to have storage. In the UK, the NHS, the hospitals are old and they're really full, so actually having storage for the dry shippers that we were talking about before is actually quite challenging. Your supply chain provider, and if we use, you know, Simon, we work with Simon and the initiative that I'm going on to talk about, you have to schedule with them. You have to be on the ball because they have to deliver the product, they have to deliver the apheresis material and equipment, they have to develop that, they have to prepare it, they have to have time for storage for product and for the equipment, and they have to interface with a data service, which I'll come back to, and they have to be involved with the scheduling as well. And then you've got the manufacturer, they have to be involved with the scheduling, they've got the storage, you've got the batch release and all of these things. And what I've heard again and again from the manufacturers is this is the first time ever that they've really had to work with the clinicians, with the nurses, and it's taken them into the transplant arena quite often, and it's an unusual way of working for them. And Phil talked about data. All of this really needs a really tight control of data, and it needs to be standardized. This data needs to be able to be transferred, whether it be between a manufacturer, between clinical sites, between the logistics. So that is critical, and the integrity of the data is crucial. But more importantly, the ability to be transferred across many, many different platforms is absolutely critical. And what do we have? We've got a really immature supply chain because it is complex. Just now, we talk about the white glove delivery. That's fine just now, but it has to develop. You have to have tracking and tracing. These products have to be traced for up to 30 years. If it's in the EU, I know in the US it's 15 years. And we talked about the chain of custody and chain of identity. That has to be maintained. So the data information and collection and the communication of that is absolutely key. And this isn't my slide, I've taken it from Cryoport, and I've just seen Rob walk in the room, so I can thank Cryoport for it, I think. And what it is, I don't want to spend too much time on it, and it's a wee bit out of date now, but what you've got at the middle of each of the circles is the licensed product, and as you go out towards the periphery, you've got clinical trial products. And all I really want to show is there's loads of different products, there's loads of different product types, and they're doing a whole load of different indications. So it's complex. So the data information collection we were talking about, you have to be considering all of this. And I've also got a projection of numbers, but this is a UK projection which we did. In the UK, we're treating just now about 200 to 300, 400 people, most of them in clinical trial. And I projected that over the next 10 years that we've got in the UK to 10,000 patient treatments. I actually think it's probably a wee bit of an underestimate now, but you can see that we're going from the white glove delivery to having to have routine delivery. So how do we go about that? So in the UK, the government has invested £30 million into the setting up of something that we're calling the Advanced Therapy Treatment Centre Network. And that it basically spans the country, it spans the UK. There's 3 centres, there's one called Northern Alliance, and that's all of Scotland and the north of England. There's IMATCH, which is isolated to one particular city, Manchester, and then there's Midlands and Wales, which covers all of Wales and the middle of England. We're involved in it, we're the coordinator of the network, and the aim of the network is to develop systems, and this is where the first and last 100 yards comes in, to develop systems for the routine delivery of these. So the routine collection of apheresis, the routine delivery of the product. We're working, it's a collaboration, I think this is key and this is why it's working. We're working with the NHS, so the hospitals, we're working with the academics, we're working with industry, well, Courier's part of it, GE through Asymptote are part of it. We've got Autolus, we've got a number of manufacturers involved as well. So by working together, we're looking to develop the systems together. And I think that's absolutely crucial for this, because if you have a developer developing something and then chucking it into the healthcare system, it simply doesn't work. It needs to be done in collaboration. There's about 35 projects, partners involved, and there's about 200 to 300 people from the NHS involved in it. So it's actually quite a sizable initiative. And we're also working with the London group, because a lot of the therapies that are developed out of the UK are developed from the universities in London. So we're also working with them. So we're trying to produce a coordinated system for the whole of the UK to facilitate the adoption of these products. So what have we done today? It's been up and running for about 18 months. And we've done quite a lot, but picked up a couple of things. And I'm going to focus on what Simon has asked me to talk about. So in terms of industry engagement, we've got an industry advisory group which has got, I think it's got about 50 members now, from very small SMEs to very large pharma. And we're working with them to see what the issues are, what are the bottlenecks, how do we start to try to solve them. We have a knowledge sharing platform, so we're developing SOPs, validation, risk assessments, GMO, gene therapy risk assessments, and we're putting them on a platform that anyone can pull off. It could be our industry partners, it could be the academics, it could be the NHS. Pharmacy, we found that the pharmacies actually in the UK weren't quite ready for these products. They didn't have the facilities, but they also didn't have the staff who really understood the products. So we're working really closely with them. We're working with them and the cell therapy labs to upscale the pharmacists while working with the cell therapy labs. Supply chain we're spending an awful lot of time on. We're looking at how do you actually understand where to pick the apheresis material up from, and how actually do you know where to deliver it back to, which sounds really simple, but actually if you've got something worth about £1 or £2 million and it goes to the wrong part of the hospital, which might actually be in a different part of the city in the UK, that's not going to be good for your product. So we're trying to work out the best way to ensure that the product gets to the right place at the right time. We're looking to increase the number of clinical trials, and we've already, in the 18 months that we've been in operation, seen a 5-fold increase in the amount of ATMP trials, or advanced therapy trials, in the UK. We're training the NHS, so we're training all of the NHS who might be touching these advanced therapies, which is quite a big ask, but it's going well so far. And we've actually started the world first MSc in ATMPs, and that's been run out of Manchester. Health economics I'm not going to talk about just now, but we're also looking at model payment systems and how do you actually manage those and how do you manage them in the healthcare system. Governance is actually really important, because what you have with these products, if you have teams having to work together that haven't ever had to work together before. So we're building multidisciplinary teams that are actually providing the governance and the policy for how we implement these products into the hospitals. And then standardisation I've left to last, because I actually think it's one of the most important things we're doing. We're looking at standardisation of so many things. We're looking at standardisation of apheresis collections, standardisation of the information that you might need to transfer between the hospitals and the manufacturers, standardisation of supply chains. So that is a critical piece of work that we're doing. And what do we have planned for the next 6 months? And this is my last slide. Again, we're going to keep on training up the pharmacy. We're going to standardise the pharmacy, and we've already got about 200 pharmacists involved, looking at training materials for them. Logistics we're spending a lot of time on. I think it's crucial. So we're spending time looking at the chain of identity. How do we keep cold chain processing? How do we ensure that the data is transferred to the right people at the right time? I talked about the standardisation of the starting material. We're also looking at patient-reported outcomes using devices, looking at training, as I explained, and actually patient engagement, which nobody's talked about today, but I think it's crucial, because these therapies actually, they're so dependent on the patient carers and on the patients who are really, really well informed. So we have to ensure that the patients and what we're developing in the NHS is suitable for them and the carers, as well as the treating clinicians and the manufacturers. And the aim of the whole process is obviously to increase access. So the first 100 yards and the last 100 yards, we're trying to make them as smooth as possible. And then hopefully through that, we're going to increase the patient access. So on that, I think I can leave.
Thanks, Jackie. Phil, come take your pick. Wherever I'm going to stand here. So thanks for everyone. It occurred to me just while everybody sits down, there was a quote from Jeff Walsh from Bluebird. I'm just trying to see if he's in the audience. And he said last year at this event, to describe what we're doing, he said, "This expletive is hard," which I think just sort of sums up exactly what we're trying to do here. So I warned you all at the beginning that this workshop only works if people ask questions. I have a load of stuff, but it would be better if it was interactive from you guys. So is anybody brave enough to kickstart, or do I have to do it? No?
Of course.
Go on, Rob.
So your great presentations. Do I need this? So great presentations showing what all of the issues are, but the journey of 1,000 miles starts with that first step. And as I'm listening to all of you, I'm wondering what that first step is. And I was thinking that from the therapy point of view and from all that you're tracking, the first step is gathering information. The more information you have, the more you know if you're doing something right or something wrong. And is the first step really to digitize, to say that what I want to do now and integrate as soon as possible as a therapy provider, as an early biotech company, is start to understand what I'm doing and understand where this is happening outside of my laboratory, outside of my process, in through logistics and having that common platform. Is that the first step? What is the first step?
So basically, is digitization the answer? Is the question?
Maybe not the answer,
Yeah.
but, what ties this together? Because there's standardization. If I'm a service provider, I have a number of different customers. I'm working in a hospital. I'm working in all these different areas. The common theme is, do I know what I'm doing?
Yeah.
Do you want to
Maybe I'll
take that,
start.
Phil?
I think yes. The first step is sort of starting to move away from. Let me back up a little. I think the first step is to do the risk assessment. Where are the challenges being introduced into the process? And you articulated very nicely that one of the challenges is we are paper-based. Papers get lost. Papers get mistranscribed. They get, wet. They get damaged. So I think. So the first question is to ask is, well, is there a better way, or what are the problems? And I think you said thank you. We've done a great job doing that, no? But more importantly, I think the first step is to start to understand how a digital solution might be part of the answer. But it comes down to really articulating, identify what are you solving for? What are you trying to fix?
Yeah, I agree. Data is crucial. Whatever, whether it be batch records, whether it be communication, and digitizing it is absolutely what we need to do. But it's so complex. If I used to work in the blood service in Scotland, and the blood service in Scotland couldn't even speak to the blood service in England sometimes, because the digitization and the digital platform was different. So yes, we do, but it's not a simple thing. But we do need to start the planning now. And I think even before we start the planning, is we need to start working together, and the industrial partners actually have to drive this, I think, to start standardising what you might want, how you might want to collect the information so that it can be transferred across multiple platforms. So I think, yes, it is the first step, but maybe even before that step is everyone getting together and coming up with a common idea.
Yeah, I think certainly agree. I think we all talk about that. We're in a great industry where we all tend to try to collaborate and be open with each other. And again, we're going to have to continue to go down that path. But I wonder also the challenge of starting, you want to start this as early as possible, but how do we do that from a development standpoint? When you get ready to go into your first clinical trials, is that already too late? When that translation happens, it all sounds really good, but we're challenged with it every day. Whether someone comes in for a research use only product, but then eventually you're going to want to go to a GMP product for clinicals, and there's always a challenge to connect those dots. And so we want to do this early, but how do we get there from your standpoint?
Yeah, maybe I'll extend my comment earlier. Maybe even upstream of that is training and educating. Why is it important?
Yeah.
Why do you collect? Why do you want to move to electronics? But I think there's a component just getting people familiar with, okay, why should we do this?
I think there's also something else. So I think digitalization is at least part of the answer, because if everybody's collecting the same data point, if you could share it, that must help. But the challenge in advance of that, so we get asked to ship a vial this big, bags this big, this size. Everybody's making it different. Some of them manifolding them up, different temperatures. So then you say, well, do we have to attach RFIDs to it, or are we going to barcode it, or are we going to QR code it? How are we going to digitize it? Some people are integrating with Chronicle or Vanek or Traxel or Homebrew. So actually, digitalization is the answer, but actually it then just moves you to a next phase of questions of how do you do it? Keith?
You could just throw that right there. You go.
Yeah.
So getting to a state of a common standard is really important. Of course, the blood services kicked this off in the 1990s with ISB T128, which is now and has been for a long time, the recognised global standard for transmission, labelling, and transfer of information for blood tissue, cells, human milk, the whole 9 yards. So I would imagine that our industry needs to get to that point. Is there any thoughts about either creating an international standards group for this purpose or leveraging ISB T128?
Do you want to take that one, Jackie?
Yes to both of those questions. So in terms of ISB T128, it's something that I myself have been advocating for a long time. Those of us who were involved with the pain of it becoming standard, it took a while, so I think we need to start working on it sooner rather than later. And through the standards coordinating body and the AABB, they are looking at a standardisation along those lines. There's a certain amount of standardisation in the EU because of the single European code that you have to apply to apheresis material, but that's European driven, so I think an international standard for the cell therapies is absolutely the way we need to go. And we were talking only last week that we do need to set up truly an international industry group who will start looking at this and bringing in the blood services who have already done it. Don't reinvent the wheel.
Anybody want to add to that?
So that's a desire, but how? Because there has to be. It can't ever be done organically from the ground up.
It's through the standards coordinating body are looking at it.
Anybody else? So I wanted to ask, Dominic, you brought up, well, you had two questions. One was with numerous logistics providers, how do you integrate effectively? That's really simple, you just use us, so we'll cover that one.
See, you're welcome.
So that standardises that straight away. But I was actually curious, you were talking about donors coming up, and we've always had to deal with people being sick or whatever, but what actually happens when the donor doesn't arrive?
Well, we don't call them back, no. So that goes back to the challenges of, again, just organizing everything and then making sure you've got everything in place when that happens. I mean, it's inevitable. It does happen for one reason or another because everybody's got something going on in their lives. So from our standpoint, it's not easy, but it's relatively straightforward. You have backup donors and backup donors in place, so if one isn't going to make it, again, you're constantly on a call with them to make sure they're going to show up. If they're not going to show up, you have the backup in place. But that's not always there. And so if it doesn't happen, or if the collection doesn't happen on time, the window is usually very small. From a pickup time, where we're located here on the West Coast, if you're going to try to get something to international, it's got to be picked up in a very short window. And if that doesn't happen, and that's where having better communication, better systems in place that aren't manual a lot of times, where you're having to call up somebody and say, hey, this is going to happen, we're calling up World Courier. It has to be much more streamlined and automated so that these, when they do come up, we have a solution that happens quickly and everybody's notified, because that's another big challenge. We get notified, but nobody else does. And that's not even talking about the actual manufacturer who's waiting for that product. By the time they get notified, it might be that next day when they were waiting for the product, and because it's in Europe and we're on the West Coast, so the time frames all get challenged. So it's again, it goes back to trying to figure out a platform that allows for all of us to be connected from start to finish.
Yeah, so I'd like to come back to machine learning in a sec, but actually one question following up from that is, as the industry grows, I mean, many of us were here when this was, a couple of hundred people, and we've now got 1,000 in this huge hotel. What's the point where we run out of healthy donors? It must happen at some point if everybody wants their donor and they want to keep them.
Yeah.
When does the industry run out of raw material?
That is a really good question. Security of supply, right? Well,
Yeah.
luckily everybody keeps procreating,
Right.
so we should be in good shape there. But
Exactly.
you know, it is a good question that we ask internally. You know, and that's why we're trying to also get ahead of, you know, doing the best you can to get ahead of what that looks like. You know, everybody's always trying to understand what is the appropriate amount of times that, or frequency that you can collect.
Yeah.
You know, and that's different depending on the type of collection. And then how do you continually broaden your donor base to do that? And I think that it is a critical challenge, and we're able to manage it today with ourselves and many others that are out there like us that are doing that. And we've got a number of different networks, but there becomes a challenge too because of the differences from the U.S. versus what's allowed in Europe. You know, so, and then other countries as to who has access to donors and who can donate. So I think that also is, again, back to standardizing and coming up with, you know, future opportunities to do this.
Okay.
Is that your tagline? We're running out of donors, we'll make more.
Working on it with the marketing team.
Okay.
I'd love to see the tagline on that. So moving from a challenge what I hadn't even thought of.
There's a question.
Sorry, is that cool?
How well do donors understand what they're donating for?
It's a good question. I mean, there's obviously very strict consents that are in place that are followed, and this goes all the way back through the blood collection and you know, through AABB and their standard collection forms and everything that they have, and consent forms. But to your point, though, how well do they really understand? I mean, this is where it gets into a challenge from a GMP standpoint and a clinical use of the material. You know, most everybody is very willing, of course, to donate their product. You know, they know that it's going for something critical and important. Oftentimes it's for research. The challenge comes into when it's for an allogeneic product that becomes a product that's worth, you know, a lot of money in a sense. Somebody's commercializing that. And again, they're consented, they sign off, and they are, from just watching our own team go through these questions with donors, we make it very apparent, and as apparent as you can, that, hey, this product could be X or it could be going to this. And so far to date, I don't think anybody has necessarily said no, but you know, I hate to say, when you look at the donors and who are donating, you know, there's certainly a level of education as well that goes into all this, and so I don't think it's going to happen. I hope it doesn't happen, but someday when the industry gets really large and there's lots of commercial success for allogeneic products, someday somebody's going to come back and say, wait, isn't that my cell that you used or my cells you used? And well, you signed off on it, but I didn't really understand what I was signing off on. So your point is very valid. It's hopefully we're safe, but time will tell.
Jackie, do you want to add to that from a
Yeah,
UK perspective?
I think I agree with what you said, but I also think there's other considerations as well, not just the commercial. I think there's also the ethical questions that you have to ask, because if you're making an IPS bank and you're doing extensive genetic testing on that, what do you do if you come across an abnormality? What do you do if you come across an abnormality that actually indicates that there could be a hereditary disease? You know, what do you do with that donor? And I think we spent quite a lot of time in the UK talking about it, and there was some guidance through a UK organisation, SABTO. And I think that's more crucial than just whether they're going to come back and say, can get some money. I think understanding whether that donor wants to know if you come across an abnormality, and what is your ethical responsibility of telling them regardless. So I don't think the donors fully understand that unless they're properly consented.
Yeah, that's an excellent point, Jackie. I mean, you brought that up and you just reminded me of that. It's from our standpoint, when we get asked by a client, you know, to do a particular test, that if it's not part of our standard testing, we have a challenge with that, because, well, we know that from our standpoint, from a business standpoint, which isn't necessarily the right reason, but we don't necessarily want the results. We don't want to know those results because, you know, we're not requiring it. And so we defer to say, look, if you want this test done, we will send that sample to you and you can test for it, but we don't want to know what that information is. And again, it's to your point, it's an ethical question and a challenge moving forward.
So just moving back, I'm curious about the machine learning piece. So Phil, we were talking before about data. So for example, we've been shipping advanced therapy since 2005. We know all of the shipping lanes, best flights, worst flights, who's delayed, everything. So in theory, we could plug all that data together and start churning out optimization, which would automatically communicate, and then Terminator would take over. But what are the thought processes going on for sort of automating manufacture and machine learning within that?
Well, I'll divide those things into two elements, right? Machine learning, I think there's two people who love the concept of machine learning. It's mathematicians, and then it's people trying to raise money in a startup company. You say machine learning, it brings people running, right? So machine learning, the challenge with machine learning is yes, we collect a lot of data, but we don't necessarily curate that data in a meaningful way that we can sort of extrapolate or collect information. And then that gets into the next level of machine learning and artificial kind of the collection, looking for patterns, looking for meaning in the data to use that meaning to then inform decisions downstream. So that is where, I won't say, well, I'll say, I think we're still years away from getting that level of sophistication of understanding of the data before we can make meaningful inferences from it. The second part of your question, or first part really, is the part about automation and how do we start to think about automation. And my first caution there is a rush to automation because we think it's going to be the cure-all panacea for all that woes us in the manufacturing environment. The challenge with automation is, what are you automating? So I'd recommend people focus a lot on, take out process steps, simplify, remove complexity in the process, then start thinking about automation. Because if you go back to your analogy of Henry Ford and the Model T, if we applied robotics the way some people are thinking about it today, he would have one robot that builds the car from front to back. And we've learned over the years that doesn't make a lot of sense. You start to functionalize and break it down into all the process steps, and then you can start to think about automating. So that's the short answer would be, look at areas that are sort of buttoned up and understood, and then we can start to apply automation principles to that. Then we can collect the data and then start to inform the next process. But it's getting the connectivity across the entire workflow, and that level of complete automation is still a few years away.
So taking it in bite-sized chunks.
Yeah,
What you're saying,
I think you
really.
have to.
And automate what we understand.
Pick the areas that are stable enough in manufacturing. That's not likely to evolve in the next 2 or 3 years in your process, and that's a starting point.
Okay, thank you. Jackie, I wanted to go back to something you said, that the manufacturing, the clinical and the logistics teams are all involved in scheduling. And I was sat there thinking, well, quite often we just get a phone call and say, can you move this now? It's ready.
Well, that's you involved.
So how much, and I have a rant quite regularly, if you've got a 10-day manufacturing process, why book the shipment on day 9? Why not do it day 1 and everybody can be sort of planning in advance? I mean, how much is that involvement actually happening and that integration and collaboration, and how much is it just throw it over the wall?
No, I would agree. I think just now it is so over the wall, and I think the couriers are seen as a courier rather than part of the process, and I think we need to move to be more part of the process. I know that and you've got some stuff up there about shipping times and stuff like that. There's a lot of information that people can draw upon from yourselves, and they should. The flip side of that is the clinical setting, they're pretty busy, and they're busy treating patients, and actually it's quite far down their list of things to think about the booking. But I think if we automated and digitised, then it would all flow through anyway. The apheresis would be booked. You would be informed of that. The manufacturer would be informed of it. So it would flow through, and I think that's where we need to get to. So you actually are knowing about it from even before the process starts, and you can start, not just World Courier, any courier can start to plan. And I know that you've told me that actually you get 15-minute slots to go pick stuff up, which is just ridiculous. People shouldn't be giving you 15-minute slots, and you need to be working with the manufacturers to make that.
Yeah, well, thank you. I'll quote you on that one. Sorry,
Yeah, I was going to extend that. It's maybe a shameless plug for the collaboration between World Courier and GE. The Chronicle software was designed largely around shipping and moving materials around, and it plugs right in with you guys, and sort of you guys should be informed when the process is moved to a certain point and should automatically schedule the pickup and the shipping.
Oh, it'd be fantastic. And I think also, when you know it's being picked up, it can also schedule the manufacturing slot or the clinical
exactly.
slot, or, there's actually that integration.
I've got an actual question to you, Simon, and actually probably everybody in here is, we talked about this a little bit the other day. When you have this product, and you hand it off to the courier or wherever that's going, who owns that product at that point? Because on our side,
It even gets a fanfare. Thank you.
and it's really an open-ended question in a sense, because we have our own, I think everybody's got their own opinion as to, in the end, we all own it, certainly, but there's a kind of an understanding. So with our respect, if we hand off the product to World Courier and they take it, we generally say it's out of our hands. It's not our product at that point. We can't control that process, but we still feel very responsible for that product to make sure that it gets to where it needs to go. But I think there's an underlying challenge as to who has the ownership from each of these steps down the process.
I guess it depends if you're asking a lawyer or an accountant,
Yeah.
right?
That's why hopefully
Let's avoid the legal definition
Yes,
of that.
exactly, that's right.
That is a rabbit hole we don't want to go down.
for sure.
But I think actually everybody owns it. Really, so the analogy I used the other day is when you take your car in for a service and you come back at 4 o'clock and they go, oh, sorry, there was a bit missing, the manufacturer let us down. Actually, you're upset at the mechanic, not the manufacturer down the line. So if we've picked it up, it's our fault if the plane doesn't take off. We need to keep control of that. But it's also your fault because you're serving the client, and it's the therapy developer's fault because they haven't got their product. So I think that's the crux of all of this, of how do we work together and how do we integrate that. So it's actually, this is a team sport. I think it's probably the answer. I'll equate it to the rugby. There's a World Cup going on at the moment and you need a scrum, and if all 8 people aren't pushing at the same time, you get shoved backwards and it's very painful. So we don't want to go there. So anybody, anything they want to throw in? Yes.
So you mentioned better integration visibility is great, but we've honestly been burned by that because you give people visibility, they plan, and in most cases, a lot of the partners here aren't that flexible yet. So you have a patient who shows up, you're still stuck into the manufacturing slot.
Yeah.
They have parts booked, they've bought their raw materials, they've started whatever. And so it's a catch-22, and so we're stuck in a situation where we're keeping World Courier on a hairpin trigger because you can respond to that because our other partners can't handle the flexibility required with better visibility, but not necessarily a sensor.
Yeah,
So I don't know how we
Yeah.
manage that as companies because it should be team sport, but honestly it's not. And ultimately we're one stuck with the patients, we're stuck with the costs, and trying to figure out sort of how we do it in a more balanced, visible way.
I think it's a very difficult question because you've got a manufacturing slot at one end and you want to reserve it and you want it available, but you don't want to be paying for it whilst you're waiting. You've got a clinical slot at one end and the patient may or may not be well enough. Never mind whether a healthy donor toes up, they just may not be physically able to do it, and then you miss your flight and then everything knocks through. But for me, it's communication and just sharing that information. Digitalization we spoke about, you could automate it, but life doesn't work that easily. We've got people in the supply chain. So it's communicating and sharing that information. So you can understand the flex, and maybe they can knock the manufacturing slot back an hour. Maybe they could do the apheresis earlier to hit a flight. But it's that communication piece. I don't know if you guys have got anything.
Yeah, I would say absolutely. The one layer atop of that I'd add, it would be the standardization. I mean, imagine a future where there's capacity for manufacturing around the world. A lot of companies come in and say, I need to block any other access to this suite because I need to know that suite's available for me when I do the manufacturing. So imagine a future where, okay, you're full, but there's capacity in Iowa. Let's send our product to Iowa. They use the same QMS system, the same BBR, so everything is the same around the world. I can ship to wherever there's excess capacity. You get the Uber model of just moving product and wherever there's excess capacity, you use it. we were a long way from that. Now, part of that's a regulatory issue, right? The ability to sort of change and use different manufacturing companies or platforms. Part of that is the lack of standards, so everybody's using different. And your product is architected around whatever it is you do. A lot of people define their product by the differences and the IP that goes into making that product. So if we start to point at the challenges and the differences and find ways then to sort of harmonize and standardize methodologies, capacities, facilities, et cetera, we'll make a step in that direction. Because then there's plenty of capacity, we just have to orchestrate it a little bit better. So that's the other element I'd add to it.
Jackie, did you want to add? Or?
No,
you're just nodding and grinning. Has anybody got anything? Sorry, at the back there.
Yeah, just curious about any ways that we can standardize the apheresis collection. Because again, every sponsor has their own parameters that they want to collect the product, and how do we overall improve the quality of this collection? All these built-in variability between the sponsors.
I'd say clone donors.
Yeah. So Dominic, that is all yours. That's
That's why we try to utilize, twins and triplets.
Exactly.
No, I mean, I think there's two different ways to look at it. Certainly, there's the donor side of it, but from a patient standpoint, which is really where the bulk of this is right now, from an autologous CAR T standpoint, there's a lot of initiatives out there to standardize the collections at the clinic, which I think is really a well worthwhile plan forward. I mean, we have to get some level of standardization. How much can you standardize, though? Because, again, it's a sick patient. You can standardize parameters, but, just looking back at what we do internally to do collections on donors, certainly it's much easier to do a healthy donor, but even still, the machines that are out there, the devices like the Optias and so forth that are commonly used, they have set standard programs, but those programs aren't perfect. So you have to, if you really want the cleanest, highest quality product, you have to override the system. But then that gets away from standardizing. So it depends on, we all have to agree on what's okay, what's acceptable. Because if we don't override that process, then the collection product that we get might not be as many cells as the client wanted, or it has too much contamination. So our job is to get the best product to them, which goes outside of standardizing. Imagine it's clearly a challenge from a patient standpoint.
Yeah.
How do you do you override, or you just kind of follow the status quo? And you could have gotten more cells, which is very critical to the process, but we wanted to standardize. So it's a great idea. I think we all have to continue to work together to figure out what that looks like.
Jackie?
Yeah, if I can add to it, part of the ATTCs in the UK, we've got a really quite a big group looking at standardisation of apheresis. And with my background from blood transfusion, I do think that we're in a lucky position in the UK because we only use Terumos. So the whole of the UK use one apheresis machine. So we're actually ideally placed in that respect. So we're looking at what's required by JC/FACT, what's a regulatory requirement. We're then looking at best practice. We're looking at what's most commonly used to develop, for a particular apheresis, whether you want an MNC and whatever. And then trying to come up with like an 80% convergence of what's required. And then thereafter, there will be variability. Manufacturers will, but what we're also doing, and I think it's really important, and I would appeal to all industrial players here, is listen to your apheresis units. They know this stuff better than you. So listen to what they advise. Don't tell them what you want, ask them what you should have. And we're spending a lot of time doing that and educating. We want to educate the industry as well. So, but we're not doing it in isolation, so we're doing that in the UK, but we're also speaking with people like Be The Match, who are doing, looking at things similarly over here. Because what we don't want is to have a UK standard that doesn't flow across. So, but I think it's a good start, and then we're going to start working internationally. But I actually think it's easier to standardise, and I think sometimes you do just need to do another, keep the donor on for longer. You're still getting the product that you need, and that's the product you need, and that's a standardisation, rather than you can have them on to full whole blood volumes and then take them off. We need to get away from that type of mentality. It's the product that you want rather than how you get there.
Yeah, I mean, it's completely, I mean, in the end, we can get there and we will get there. It's just, we're still young. I mean, the industry is still really young comparatively, so we'll get to the level of standardization, and it may not be the exact, as you said, it's piecing it out again, and as Phil had mentioned, you know, looking at different parts of it that can be standardized. It may not be a one-size-fits-all, but different platforms become standardized, and again, standardizing between regions.
Yeah.
Getting that under control is going to really help.
I'd also throw in that trying to standardize the last 100 meters. So do you deliver to pharmacy, to the cell bank, to the blood bank, to stores? Where does it go, and how do you manage that within the clinic, just that sort of practical aspects of moving things around hospital corridors? So I've got two questions to finish, one sort of reflective and one looking forward. So Phil, if we start with you, if you could have done anything differently, what should the industry have done to be in a better place than they are now?
That's a really good question, clearly. Right?
Thank you.
Yes, exactly. Probably a number of things. I think that the appreciation, and this is something that when I was in my prior life at Lonza, you know, people would come in with a translated product from the clinical centers and then try to weren't really fully understanding scalability, the number of patients they deal. I would say that one thing that would be really, really useful is sort of pushing an appreciation for what it will take to industrialize this, all of these types of complicated therapies, and provide that guidance earlier into the clinical setting so people have a better appreciation of what even it means. What does supply chain mean? What is, you know, treating 100,000 patients going to need to look like? Because we spend a lot of time and energy in this industry redesigning a manufacturing process to make it manufacturable as opposed to making it a clinical product.
Okay. So that vision for the future.
Yeah.
Jackie?
I think if we were to go back 10, 15 years, I think standardization. And with data and with everything, you know, if you think about if we'd all started working together early on, a lot of the issues and a lot of the barriers that we have now, we wouldn't have. If you think about semiconductors in Japan and how they decided to do it all the same, and it became a whole lot easier. So it means that not only does it make the early steps, but it also makes the ability to progress and progress quickly much easier. So I think standardization.
Great.
Yeah, it's easy to say, but really hard to do, and it's slow down. You know, I keep telling myself to do that too in some of the different projects, but if we slow down, we can gather the appropriate information, especially now, just, you know, going and talking to each of the respective parties from start to finish that haven't always been included in this process, and you know, maybe we can learn from that.
Okay, so I was almost playing back to the collaboration piece again.
Yep.
So to finish then, if you had a magic wand and you can do anything going forwards to make the industry better, what would you do? Phil?
Oh, okay, sure, yeah, no.
You were dreaming there.
Oh, yeah, no, I'm thinking through. I'd probably put everybody on Chronicle, right? No, seriously, no, I would absolutely push for, you know, data collection and real emphasis on. I would love to have a really good understanding of the biological consequences of pathways and all that, so we could be better engineers of cells, and then we could use that information to design better manufacturing processes.
Okay, information. Jackie?
Curated data.
It's information as well. I think making the clinical setting suitable for the adoption of these therapies. And part of that actually is also having, my wish would be to have a registry that everybody could feed into internationally that could be fit for purpose for clinical reasons, for regulatory and for reimbursement.
Okay, a small wish.
Is that all?
Dominic?
It's obvious. It's, you know, for allogeneics, it's a super donor. You know, one donor suits all. I think it's exactly what these guys have said, but you know, again, it's an education. You know, more education that's out there from a collaborative standpoint, and having access to more, you know, accessible resources so that everybody that's coming on board that's really involved in this, and we're moving so fast, it's having, you know, more educational resources that can allow people to have the information sooner than later so that we can develop the scalable processes.
Okay. So to summarize then, I think it was collaboration, and collaboration is what came out. So thank you all for attending.